Alright, hello to another video. So last video we were talking about the carbon cycle and the way carbon actually propagates through an ecosystem from the main from the main atmospheric and, water reservoir through an ecosystem and back to that same reservoir. So right now we're going to be doing a similar thing with the nitrogen cycle. Of course, nitrogen is a necessary organic atom, or is a necessary atom in the production of organic molecules needed to actually sustain life in plants, and by extension, a lot of other, important organic molecules that we find in consumers and other organisms like ourselves. So we're gonna start off by drawing our main source of nitrogen, which is always typically found in the atmosphere. So I'm going to really quickly jot that down. So atmospheric N2. So with atmospheric N2, N2 isn't inherently a greenhouse gas, but nitrous oxide is, and I thought that was just something interesting to point out. So where do we go with this atmospheric nitrogen? Well, the first step of this nitrogen cycle is that nitrogen must be first kind of assimilated or kind of converted to ammonia. And this is known as nitrogen fixation. Now, nitrogen fixation can actually occur in two ways. The first way it happens is in an artificial process. This artificial process is known as the Haber process. The Haber process is used in a lot of industries to synthetically create NH3, and it works by making an equation of N2 plus H2 and reacting these two essentially elements or atoms using a metal catalyst. But that's more chemistry, and I won't get too much into it. The other way NH3 is kind of fixated, so let me actually put that word here. Fixated is the key word. The other way ammonia is fixated into ammonia, or nitrogen is fixated into ammonia, sorry, I'm getting this mixed up, is naturally, by nitrogen fixing bacteria. And there are actually two nitrogen fixing bacteria I will talk about, but the requisite kind of conditions of any bacteria in order to actually produce NH3 from atmospheric N2 is that they must have an enzyme called nitrogenase, and it just allows them to process N2 in a way it can create ammonia. But the two central organisms or bacteria that play key here are, first of all, Azotobacter, which is free living meaning it lives independently. And then Rhizobium is not free living, and that just means it always occurs in a mutualistic symbiotic relationship with legumes. And the way this works is that, very, very briefly, you have Rhizobium and you have legumes, and these legumes essentially have nodules, and then these Rhizobium come and invade these nodules, and from those nodules can essentially assimilate nitrogen or N2. Sorry, I was about to say nitrogen dioxide, but they essentially assimilate N2 and create NH3. Now, this is a symbiotic relationship because the Rhizobium, of course, benefits from being in the nodule, and also by essentially taking residual carbohydrates from the legume. And then the legume, of course, profits because it's getting NH3, which it can then use to actually assimilate protein in its tissue. So let's actually draw that flux. That's an important flux. We'll call this assimilation. Assimilation of NH3. And we're going to say that this is going to the protein, or plant protein. I will do this in blue to keep consistent. Plant protein. Now, before we go any further, we now have two things we need to look at. we have the NH3 reservoir, and we have the plant protein reservoir. So I'm going to go and stick to the plant protein reservoir, then I'll loop back to the NH3. So what happens to the plant protein? Well, there's not much else we can go from here. So plants just end up dying, and through death we now have, again, dead/decaying or already decayed organic material. So I'm going to call this organic matter. Okay, I'm going to call this organic matter. And then this begs the question, well, what happens to this organic matter? Well, by, bacteria and by decay via saprotrophic bacteria, through external digestion, the nitrogen, the NH3 that is found in this organic matter is then kind of recycled back into the soil. So that process is just going to be represented with this flux. Then we can just call this decay. Decay. So now we're kind of done with this mini cycle here from NH3 to plant protein back to NH3. So what else can actually happen to this ammonia here? Well, in the process of nitrogen fixation, there's actually a couple more steps. And from NH3, this NH3 actually then becomes nitrites, NO2 minus, by a bacteria known as Nitrosomonas. I usually get that mixed up with the other bacteria I'll talk about in a bit, but this is done by Nitrosomonas. And this process here is known as nitrification. Remember, it's called nitrification because it forms nitrite. Okay? So from NO2, we can then produce NO3, or nitrates. So we get NO3 minus. And this is done by bacteria known as Nitrobacter. That was the bacteria I was about to mix up. This is done by Nitrobacter. And this is still the second step of nitrification, so that's exactly what we'll call it. So now we have kind of the NO3. Well, NO3 can actually be used or two things can essentially happen to it. NO3, nitrate, just like ammonia, can be uptaken by plants and help form plant protein. So I'm going to draw this flux right here. I'm just going to call this assimilation. Because it's effectively doing the same thing as this kind of flux here. So we have assimilation leading back to organic matter, right? So that's good. Well, I actually missed something here, and I just remembered. Plant protein, or plants in general, plant tissues, can again be eaten through herbivory. It can be eaten by primary consumers, and those primary consumers can be eaten by higher order consumers. So I'm going to create a new flux from plant protein to consumers. And this flux I'll call here is herbivory, but really it's just any general consumption because herbivory is only between the plant and the herbivore. So that's nice. Now we have a nice complex chain going on here. Well, what actually happens to consumers? Eventually they do die, just like plants themselves die. So I'm going to draw this flux, and I'm also going to say here that this is death. Oopsie, I accidentally switched to gray. But this is also death. So now we're getting somewhere. We have the complex chain, but we didn't really address what else happens to the nitrate, because yes, it can be assimilated, but the last step to this entire chain is something known as denitrification. And denitrification is done by a bacteria primarily called Pseudomonas denitrificans, which effectively breaks down or it removes the oxygen and nitrate in a reduction reaction. So and in doing so, it creates nitrogen, atmospheric N2. It recycles back into our main reservoir. So this process here is known as denitrification. This is not very neat, my bad. But this is known as denitrification, and again, it is done by Pseudomonas denitrificans. Okay? So this is kind of what happens, and we also said that this occurs via reduction. But that's not too important. That's just kind of if you want to know. So yeah, that's pretty much the nitrogen chain. If you have any questions, feel free to ask. And yeah, thank you.